The aim of this experiment is to study whether the ingestion of dissolved glucose may differ in energy expenditure, fat and carbohydrate oxidation compared to the ingestion of dissolved sucrose. This comparison will be made between fasted and fed conditions. This leads to the research question: what are the different effects of consuming glucose or sucrose on energy expenditure, fat and carbohydrate oxidation during exercise? Before a hypothesis could be formed, a bit of biological background information was needed on the absorption of carbohydrates. Glucose can immediately be absorbed and used by the body. Sucrose, a disaccharide mostly known as table sugar, needs to be split up in a single fructose and a single glucose molecule before it can be used by the body. The absorption of carbohydrates is dependent on a few transporters, namely SGLT, GLUT2, and GLUT5 transporter. The intestinal glucose absorption is mediated by SGLT through the intestinal mucosa and exits at the GLUT2 transporter into the capilla. For fructose, the GLUT5 transporter leads the fructose into the mucosa and GLUT2 is the exit point. The breaking down of the fructose and the different absorption pathway may take more time than the immediate absorption of glucose and cost more energy expenditure, and higher fat might be oxidated. As a hypothesis, it will be expected that the person consuming sucrose will have a higher energy expenditure, higher fat and lower carbohydrate oxidation than the person consuming glucose, as sucrose probably first needs to be converted into fructose and glucose, which might take some more time. For the experiment, first the baseline is measured. This happens during the 30 minutes while resting under fasting conditions. The subjects haven't eaten anything since 4 hours prior to the experiment. Then the ingestion of 2 solutions occur. These have to be consumed within 2 minutes. Immediately after the consumption, the 2 students start cycling for 60 minutes while trying to keep the Watts level at 55. The main materials used during the experiment are 2 calorimetry machines, 2 cycloergometry, and a laptop with Excel. The 2 subjects were randomly chosen and comparable towards each other's weight, height, and fitness. For the ingestion, one test subject takes in a glucose solution which consists of 75 grams of glucose dissolved in 250 milliliters of water. The other test subject takes in a sucrose solution. The amount of 75 grams was chosen since it's a common value in multiple experiments. For example, the OGTT also used a value of 75 grams. The 250 milliliters of water was chosen since this is the amount necessary to dissolve the glucose and sucrose. To avoid any placebo effect, the substances are made unknown for both the test subjects as well as the researcher through the blinding method. During the measurement, the researchers wrote down the different values for fat and carbohydrate oxidation, for the RQ, and for the energy expenditure. The following calculations were made based upon the retrieved data. The average energy expenditure per 15 minutes were calculated both in resting mode and during cycling. The fat and carbohydrate oxidation were also calculated as an average per 15 minutes. The fat and carbohydrate oxidation had to first be converted into kilojoule per minute before it could be taken as a percentage of the energy expenditure. The fat oxidation was multiplied by 37.7 while the carbohydrate by 16.7. Then the oxidations were measured as a percentage of the energy expenditure. This was done because it gives a clear indication on how the oxidation of fat and carbohydrate changes. Furthermore, the respiratory quotient, the RQ, was calculated by dividing the produced CO2 by the consumed O2. The RQ gives an indication which substance is being oxidized. An RQ equal to 1 indicates that carbohydrates are oxidized, while an RQ close to 0.7 means that fats are being oxidized. For the RQ, averages were measured during the fasting condition, but also after feeding condition per 15 minutes. When looking at the results in percentages for the 30 minutes baseline measurement that was divided in two groups of 15 minutes in subject A and B, we notice a higher carbohydrate and a lower fat oxidation in the first 50 minutes of subject A. This changes in the last 15 minutes in which the fat oxidation increases and the carbohydrate decreases. However, for subject B, the carbohydrate and fat oxidation are almost equal during the first 15 minutes before the fat oxidation increases and the carbohydrate oxidation decreases. When we look at the RQ for the 30 minutes baseline measurement of subject A in red and subject B in blue, the RQ lowers over time in these 30 minutes, in which subject B has a more steeper decline than subject A. After the ingestion of unknown substance A by subject A and substance B by subject B for the 60-minute experimental measurement, subject A fat and carbohydrate oxidation stayed almost the same for the complete hour, while subject B fat was mainly oxidized in the first 15 minutes and decreased slowly during the remaining 45 minutes. During those 45 minutes, carbohydrate oxidation increased slowly. When we look at the RQ for the 60-minute experimental measurement, the RQ for subject A is almost stable during the first 30 minutes and increases in a small margin for 15 minutes and stabilizes in the last 15 minutes. In subject B, the RQ starts with a 0.7 and increases during the first 45 minutes until it stabilizes in the last 15 minutes, in which the RQ of subject A and B are almost equal towards each other. Fat is oxidized, it can have a higher energy expenditure percentage than carbohydrates. However, fat doesn't necessarily have more grams per minute oxidized. This is due to the fact that fat is multiplied by 37.7 to calculate the kilojoule per minute and carbohydrate with 16.7. As expected, the energy expenditure of the subject consuming glucose was more dependent on carbohydrate oxidation than fat oxidation. This can be explained by the presence of single glucose molecules that are digested and absorbed immediately. Because of this, the energy expenditure can rely on carbohydrate as long as they are available. Subject B, consuming sucrose, had an energy expenditure that mostly relied on fat oxidation in the first 30 minutes of cycling. After the first 30 minutes, the oxidation shifted from fat to carbohydrates. This confirms the literature stating that the breakdown of sucrose and the different absorption pathways may take more time. It should be taken into consideration that subject B already had a high fat oxidation during fasting in comparison to subject A. These results can also be seen when comparing the experiment RQ. Subject B relies on fat oxidation during the first 15 minutes, as can be concluded from the RQ. During the experiment, the RQ starts at a value of almost 0.7, which indicates that the substance being oxidized is fat, while subject A has an RQ of almost 0.9 since the start of the experiment, and an RQ of 1 equals the oxidation of carbohydrates. The RQ of subject B starts rising from the start and eventually reaches almost the same value as the RQ of subject A. Based on this experiment, the hypothesis the person consuming sucrose will have a higher energy expenditure, higher fat and lower carbohydrate oxidation than the person consuming glucose, as sucrose probably first needs to be converted into fructose and glucose, which might take more time, can be accepted. However, our test group is very small and evidence-based conclusions cannot be drawn. These two subjects are not representable for the whole population. Additionally, we could not measure whether the two subjects were given an equal effort during cycling. Also, differences in metabolism between subject A and B could be a confounder for the result of this experiment. That is why it is also important to measure this in further research experiments.